Hi again! So previously, we learned about the conditions required for gene therapy as well as the two ways that gene transfer can occur. Now, we'll be learning about the different gene delivery systems used in gene therapy. The single greatest challenge in gene therapy is to deliver large fragments of DNA to the correct cell and to the correct site on the large human genome without affecting the functions of other genes. Gene delivery system can either be viral mediated or non-viral mediated. Characteristics of an ideal gene delivery system would be that it has a high uptake efficiency of the normal functional allele by target cells. Secondly, it has minimal intracellular degradation during transportation of the normal functional gene to the nucleus of the target cells. And thirdly, there should be sustained expression of the normal functional gene product at a level that is able to alleviate the disease and its symptoms. So now let's move on to viral-mediated gene delivery systems. Viruses are naturally able to encapsulate and deliver the genes to human cells in a pathogenic manner. Scientists take advantage of this to insert therapeutic genes. In fact, about 75% of gene therapy protocols use viral vectors. The general procedure is as follows. First, we select a viral vector that targets only the specific cell type. Then, we disable the virus vector by removing or inactivating disease-causing genes from the viral genome. For example, genes giving the virus the ability to replicate itself must be removed so that no new viral particles are created. It also makes room for the insertion of a transgene because space in the viral capsid is very limited. Then, we insert the normal functioning non-viral genes into the virus. Lastly, we then use this virus to transfect either extracted cells ex vivo or introduce it directly into the patient using the in vivo approach. Different types of viruses are used, such as retroviruses, adenoviruses, and adeno-associated viruses. Only retroviruses are in your syllabus, but if you're interested, do read up on the other types of viruses used because it's pretty interesting. Retroviruses are able to create double-stranded DNA copies of their RNA genomes via an enzyme called reverse transcriptase, and then integrate these double-stranded DNA copies randomly into the chromosomes of host cells via another enzyme called integrase. Examples of retroviruses include the Bologna-Murine leukemia virus as well as the lentivirus, which is derived from HIV. Retroviruses are used for majority of gene therapy experiments because of their high transfection efficiency. This makes it the most established and most efficient method of introducing genes into mammalian cells. An advantage is that it is able to integrate into the human cell genome, so gene expression is stable. Integration is required, or else the introduced genetic material will be a target for nucleases in the cytoplasm or nucleus. Some disadvantages of using retroviruses is that it can only carry small pieces of DNA, smaller than 8 kb, and many human genes, even without introns, are larger than that. Secondly, the target cell has to be rapidly dividing in order for viral DNA to be incorporated into its chromosome. However, since most cells in an adult's body have fully differentiated and stopped dividing, this shrinks the amount of available cells. Why must the target cells be dividing for retroviral DNA to be integrated into the host cell genome? This is mainly because viruses need to wait for the nuclear membrane to disintegrate before it's able to introduce its genetic material. And this disintegration of the nuclear membrane occurs at times like cell division. However, lentivirus is able to infect both dividing and non-dividing cells as it can directly inject its DNA through the nuclear membrane. Another disadvantage is that integrase inserts viral DNA randomly, and this may lead to insertational mutagenesis, where a tumor suppressor gene is inactivated due to insertational inactivation, or when the viral promoter causes the overexpression of oncogenes. If that happens, that might lead to cancer. this checkpoint, you should be able to name an example of a viral-mediated gene delivery system as well as its advantages and disadvantages.